All right, good morning again, everyone. On last class, we started discussing microbial diversity. We started this conversation off looking at viruses. All right, so we're clear that there are quite a diverse range of organisms that exist. And so in our conversation of microbial diversity, we started first talking about the virus. And so we started with a characterization of the virus, and then we looked at some individual properties or properties that are specific of viruses, particularly the viral structure. And then today I want to talk a little bit about some diseases that are associated with viral infections and being able to distinguish between virions and viroids. So most importantly, on last class, we established that viruses were in fact acellular organisms. Okay, they do not exhibit characteristics that are found in cellular organisms, and that they can carry out those characteristics of life by themselves. We know that viruses are essentially parasitic in nature, right? They require a host cell. Right. So some of the text that you read, you may see the term obligate intracellular. Okay. That means that these viruses cannot survive on their own without infecting a host cell. Right. And so they are in a category of their own. All right. Other microorganisms that we'll get to later are cellular organisms, bacteria, archaea, protozoa, and fungi. All right, but today we're still talking about viruses. We said that it's more correct to refer to viruses as infectious particles, right? These infectious particles are basically going to be like little wads of genetic material, right? Which are either DNA or RNA viruses. And then they're enclosed by this protein capsid, right? We said that some of them can contain an envelope as an additional barrier of protection around that protein capsid, okay? And so we established 5 identifying characteristics of viruses on last class, being that their genomes are either DNA or RNA, excuse me, that they're unable to replicate on their own. They do not utilize normal cellular division that we see in other organisms, such as binary fission. All right, they lack metabolic or enzymatic potential. They require the machinery of the host essentially to replicate their viral genome. All right, and so we looked at some different types of viruses here, but we established that a basic structure of the virus is either it's DNA, they're a nucleic acid. It's DNA or RNA, all right? So they have genetic material or a genome that's surrounded by a protein coat, okay? So today, let's talk a little bit more about some specific types of viruses, and we'll start with the bacteriophage. Write this down. Bacteriophages are viruses that infect bacteria, right? So we said on last class that pretty much all cell types are susceptible to viral infections, right? Bacteria cells are specifically infected by viruses that we call phages, okay? A bacteriophage is a virus that infects bacteria. And so once this infection occurs of this host bacteria cell, there are a couple of things that can happen, all right? So phages can be identified or classified based on how they respond once they have infected a bacteria cell, okay? And so write this down. There are 2 types of phages based on their life cycle after infection, okay? We have virulent phages, right? These virulent phages are gonna lead to what we call a lytic life cycle, right? This lytic life cycle is ultimately going to lead to the lysis or destruction of the bacteria cell, okay? So we have virulent phages, and then we have lysogenic phages, all right? Lysogenic phages, jot this information down. These are going to be characterized by sort of a temperate life cycle, okay? Once the infection of the bacteria cell has occurred, Temperate phages are essentially going to integrate into the bacteria cell genome. All right, so it's going to integrate into this host DNA, and it's just going to sit there for a while. Okay, and then under certain circumstances, it can become this lysogenic phase can become a lytic phase and ultimately to the lysis and destruction of the bacteria cell. So bacteriophages can exhibit these two different life cycles after infection, lytic life cycle and the temperate life cycle. And I'd like you to be able to describe what's happening during both of these. And so once the bacteriophage is sort of attached to this host cell, there are some things that's happening. So let's look at these stages of replication or multiplication of a bacteriophage inside of its bacteria host. Okay, so the first step is going to be attachment. All right, in order for anything to occur here between this viral particle and this bacteria cell, this virus, this phage, has to attach. Okay, so it's going to attach to a receptor on the surface of the bacteria cell. Okay, once attachment occurs, this phage is essentially going to penetrate and inject its DNA into the bacteria cell. Okay, so there's an injection of genetic material happening, right, for this infection. And so then biosynthesis takes place, and this is when the viral gene particles are being expressed. Okay, so the production of viral particles and proteins are going to take place once inside of this bacteria cell. Okay, and basically new viral particles will begin to assemble. Okay, once these viral genes begin to be expressed, new proteins are going to be made, assembly of new viral particles will take place, and then this accumulation of new viral particles will essentially lead to the lysis of these new viral particles and the release, okay, once that cell lyses. Okay. So I want you to jot those steps down. And be familiar with what happens during this lytic life cycle of a bacteriophage infection. Okay. And so this is just a visual again of what's happening during the lytic process, right? So the first step here is attachment. So this is our bacteria cell, which is our host, right? This this bacteriophage, in order to get into cause the infection, to inject this genetic material, it needs to attach. Okay, so attachment of the phage to a cell surface receptor, right, on our bacteria cell takes place. Okay, the next step is injection of the phage DNA into the cell. Right, so we've injected our genetic material here, and then synthesis of these viral proteins must now take place. Okay, so synthesis of new phage DNA and phage protein are being made, and then the assembly of new phage particles will commence. All right, so new phage particles are going to be created. These complete phage particles are essentially going to take over this bacteria cell and ultimately lead to lysis and release of these progeny phages, and they will go on to do the same thing to another cell. So this lytic process is one that is ultimately, it can ultimately be problematic for the bacteria, for the viruses, right? Because the goal here is not to lyse all of the bacteria cells, because without the bacteria cells, these viruses cannot survive. So it's not very great for the virus, although that's what's happening. So another type of virus that we see are animal viruses, and we collectively refer to viruses that infect both humans and animals as animal viruses. All right. These animal viruses can have either DNA or RNA as their genome, right? They can be without that envelope, so they have the simple structure of the nucleic acid surrounded by this protein capsid, or they can also be enveloped, right? So some animal viruses are enveloped, right? And so we see this envelope just as an additional barrier of protection around the virus, right? And there are other proteins and so forth. These spikes that we see coming off of these envelopes are characteristic of some viruses, like the flu virus and so forth, the coronavirus. All right, so these are viruses that can infect either humans or animals. All right. The multiplication process is very similar to that we saw in bacteria cells, right? So in order for this viral infection to take place, there's got to be attachment, right? The virus attaches to a protein or a receptor on the surface of the host cell, right? Penetration, where this entire virus in this case is going to enter the host cell, all right? Uncoating, which is going to lead to the release of the nucleic acid from the coating. All right, so this is a step that's a little bit different than what we saw with the bacteriophage, right? Phages inject their genetic material. With animal viruses, there's a complete penetration of the viral particle into this host cell, right? And so uncoating has to happen before biosynthesis of these new viral particles, okay? Before these viral genes are being synthesized by the host cell machinery, it's got to uncoat. All right, and then so we create these new viral parts, pieces of the virus, right? The DNA, viral proteins, and so forth. And so we've got to go through the process of assembly again. All right, so viral assembly takes place to complete the formation of new virions, right? Virions are complete viral particles, and then the release. All right, again, these newly created virions can leave the host cell by way of lysis or budding. All right, and so this is what happens with animal viral infections, okay? Multiplication of animal viruses. I would like for you to be able to describe this process and again, distinguish how this one is a little bit different than the replication of a bacteriophage. Okay. Excuse me. And so then we also have a type of viral infection that are called latent viral infections. Jot this down. Latent viral infections are when an infected person harbors these virus cells, but they may not necessarily always express evidence of an infection, right? So these latent viral infections can lie dormant in the human cell for years, all right? And so they are usually contained by the host defenses, right? As long as you have an intact immune system, for the most part, these latent viral infections don't cause problems, but they can become active under certain circumstances of, a host weakened immune systems, things like stress, becoming sick with another disease, getting older. There are factors that will contribute to the activation of these latent viral genes at some point in your life if you are infected with the latent viral infection. Okay. And so, again, for the most part, as long as your immune system is intact, you are pretty much protected from these outbreaks, right? But in the event that you become, that you experience a weakened immune system, your immune defenses become compromised, you are at risk for outbreaks, right? So some examples of latent viral infections are, we see them in herpes viral infections. Individuals with herpes simplex 1 can have cold sores or what we call fever blisters that come and go, right? And so these outbreaks are completely, I won't say random, but again, they're due to some type of stress or a change in your defenses that leads to these outbreaks becoming active. All right. So we have these cold sores or fever blisters. All right. Excuse me. Shingles are another latent viral infections that we tend to see in the elderly. All right. Because most of us, well, depending on your age, you guys probably haven't because you've all been vaccinating your whole entire life, but elderly people who may have contracted chickenpox can later present with shingles later in life. Again, this latent viral infection can lie dormant in your body for years. And individuals with shingles, which is a very painful neurological disease, shingles can result after years after a chickenpox infection. Okay, so we typically see shingles in elderly people who, where, as you get older, your immune defenses become more and more weakened, and these viral particles can become activated once again later in life and cause shingles. All right. And this is, again, reactivation after a chickenpox infection. Another type of virus are oncogenic viruses. Okay, jot this down. By definition, these oncogenic viruses are those viruses that cause cancer. All right. There are a number of cancers, Epstein-Barr virus, that can cause cancer. Burkitt's lymphoma is caused by a virus. Our Kaposi sarcoma is caused by a virus. And so, yes, there's evidence that suggests that viruses can lead to our cells converting and becoming cancerous. Okay, we know that cancer is essentially a result of aberrant or uncontrolled cell growth, right? And so it always results from a normal cell that has undergone some type of deviation from the norm, right? So cancer causing agents, damage, injury, a number of things can contribute to the conversion of this normal cell into a cancerous cell, all right? And in this case, viruses infecting normal cells can lead to the cell becoming a cancerous cell, all right? And so for this reason, again, viruses are extremely important in terms of their role or impact on both the humans and animals, okay? And all microorganisms. I started this conversation again saying that there was no cell that was sort of immune or exempt from viral infections. Viruses can infect all cell types, all right? And then we have some other viral-like particles that we need to highlight, okay? We have viroids, which again, they're not organisms, they're not cellular organisms on their own, but these viroids are just infectious RNA particles, RNA molecules, okay? These are short single-stranded RNA molecules that can cause disease, okay? Viroids typically cause disease in plants, okay? They cause disease in plants. And then we have prions. You've probably heard about prions before. These are infectious protein molecules, all right? Prions can infect both humans and animals. Okay, and so again, as we're here looking at viruses as an important acellular organism, we have to discuss these infectious agents as well. Viroids, which are infectious RNA molecules, and prions. All right, these prions again are just some altered proteins. There's changes in the conformation of our proteins. These proteins are misfolded, okay, and it's going to confer different properties on these proteins, variants that ultimately can cause some very severe and potentially fatal diseases in humans and animals, okay? So I'm gonna stop here. I want us to talk a little bit. I want you to spend a little time with these checkpoint questions before we move on to our next category of microbial diversity. We can spend the rest of today's class. We have about 20 minutes remaining. It may take you a few more minutes to get this done, but there's a submission created for you in Blackboard. Everyone's going to work on this today independently. Your answers are due by the end of class today.